in the enteric nervous system and through paracrines and hormones. So let's think now about the rest of the story. You've heard always about how things get churned up in the stomach and acidified and turned into the chyme that will be called vomit. But what about when it gets into the intestine? There's really a surprising amount of physiology there. Let's start with the acidity of the chyme. What does that stimulate? Go. The acid stimulates the release of the hormone secretin. It acts as a hormone going to the pancreas to stimulate bicarbonate secretion. So you mostly got that right. And that's the key right there, is that the bicarbonate neutralizes the chyme. The pH might be 2 or 3 coming out of the stomach. The bicarbonate secretion has a high pH. And between the two of them, you end up with neutral chyme. That's a good thing. Secretin also goes through the bloodstream to inhibit gastric motility and acid secretion in the stomach to keep us from overdoing it with the acidity. And we saw a lot about what regulates acid secretion within the walls of the stomach through paracrines last time. We also saw last time how the enteric nervous system, including its stimulus from the vagus nerve, affects the acid secretion of the stomach, stimulating it. Here we see it inhibiting it when signaled by chyme getting into the small intestine through neurons of the enteric nervous system. I'm guessing, though I don't know for sure, that these would not be vagus nerve factors. These might be interneurons in the enteric nervous system. Question 2. What about the fat in the chyme? What does it stimulate? Next question. What do carbohydrates, namely starch, glycogen, disaccharides, and sugars, induce? Answer is GIP and GLP-1 are secreted. Let's see if I can remember. Gastric inhibitory peptide was the original meaning of GIP, but now it stands for something else. And it's like insulin. And glucagon-like peptide 1 also acts like insulin. This one named for, if I'm remembering correctly, I hope I am. Help me out, Morgan. I forgot to study this last night. The fact that it looks like glucagon, it nevertheless acts like insulin. And this one is not actually inhibitory to the stomach after all. It acts like insulin too. And I stands for insulinotropic. It stimulates the release of insulin from the pancreas. But I'm blanking on what the G stands for. So these things, carbohydrates, are going to stimulate the pancreas to release insulin, getting ready for the sugars to get taken up into the bloodstream rise in concentration and therefore be available for the tissues to use. Question 4. The pancreatic ducts and intestinal epithelium secrete bicarbonate-rich solution. Why do they? What are the transporters in the apical and basolateral membranes that do the pumping? Why does the transport also draw water? What if you have cystic fibrosis? Go. Okay, the answer is that we need bicarbonate-rich secretion secreted into the lumen of the intestine via the lumen of the pancreas, pancreatic ducts, to neutralize the chyme or buffer the acid of the chyme. So bicarbonate is transported at the apical membrane of pancreatic duct cells, as well as some of the intestinal cells, and that bicarbonate neutralizes the chyme. It's a bicarbonate chloride exchanger like we have in the red blood cells, which help us in transporting bicarbonate from the tissues and getting it in the red blood cells in the tissues and then out in the lungs where it can be, back in the tissues and, out in the tissues and back in the lungs where it can be taken in to convert to carbon dioxide. In the case of the lungs, the carbon dioxide gets out into the capillaries. Here, the carbonic anhydrase is converting carbon dioxide into bicarbonate to give us a source of bicarbonate. The chloride would build up inside the cells were it not for the CFTR, the cystic fibrosis transmembrane regulator channel, the chloride channel that when mutated doesn't let chloride out. And the effect of that is to plug up the pancreas ducts, because without the buildup of bicarbonate and chloride, you don't get as much osmotic draw on the water from the interstitial fluid of the pancreas to liquefy the chyme all the more. This negative charge draws sodium out, so you really get sodium chloride and water and bicarbonate from the effects of the chloride bicarbonate exchanger and the CFTR channel. On the other side of things, hydrogen is getting pumped out, sodium is getting pumped in with it, counterport, but pumped back out. Salt is getting pumped in, the potassium back out. So the excess chloride that's coming in from the sodium to chloride potassium transporter is available to go out the CFTR channel. So that's how the transport also draws water. It's not through the transporter, but it's through the paracellular pathway by an osmotic mechanism. And if you have cystic fibrosis, you will get clogged up pancreatic ducts. Next question. Secretin activates the pancreatic alveoli to secrete digestive enzymes. The intestinal epithelium also secretes an important enzyme that activates an enzyme from the pancreas, so it can activate the others. What are the 7 main enzymes and what macromolecules do they cleave? See if you can list them. 7 enzymes and their corresponding macromolecules of importance. Go. Okay. The pancreatic proteases include enteropeptidase in the wall, meaning on the apical membranes of luminal epithelial cells of the intestine. Cleaves trypsinogen, which is coming from the alveoli of the pancreas to make trypsin. Trypsin cleaves chymotrypsinogen from the alveoli, procarboxypeptidase, procolipase, and prophospholipase. To make chymotrypsin, carboxypeptidase, colipase, and phospholipase. Now you're going to get some clue from the words oftentimes as to what these things will then cleave. Trypsin is cleaving these enzymes, right? So it must be a protein cleaver because these enzymes are all proteins. So it would be a protease. So trypsinogen is the parent to a protease. Chymotrypsin is the trypsin in the chyme. Not that trypsin isn't in the chyme as well, but this was probably where it was originally identified, chymotrypsin. Carboxypeptidase is going to cleave peptides. Colipase is a partner in breaking down lipids. Phospholipase breaks down phospholipids. That's all pretty straightforward. And not on this diagram is amylase, which digests starch. Now there's salivary amylase and pancreatic amylase, and even, as I recall, a little bit of gastric amylase. So they all break down starch, but the salivary amylase is inhibited by the acidity of the chyme in the stomach, and it stops functioning. And it's not until we get to the duodenum and the jejunum and ileum that we get most of the amylase effect, where the starch and also starch from plants and glycogen from animals gets broken down. And they get broken down as carbohydrates into disaccharides and monosaccharides. So next question, question 6. Which monosaccharides make each of the 3 main disaccharides? And by that I mean make up each of the 3 main disaccharides. So list the 3 main disaccharides and what 2 monosaccharides they're made of. Go. Thank you, Amber, for doing that right. Meaning, reading my mind correctly. And Ryan, too, good job. And the rest of you at the beginning named the monosaccharides very well. That's good, because that's really the way the question was worded. Answer is, starch and glycogen are broken down by amylase into disaccharides, maltose, sucrose, and lactose. Lactose is named for being the milk sugar, lactation. It's the main sugar available in babies in breast milk. Sucrose is our table sugar. And maltose is two glucose molecules attached to each other. Since glycogen is made up of glucose exclusively, as I recall, it's broken down only into maltose. And maltase cleaves maltose to make two glucose molecules. Sucrase cleaves sucrose to make a glucose and a fructose. Fructose is here diagrammed as a 5-membered ring, and glucose is a 6-membered ring. And throughout the book, that's the approach, that's the symbol that the author has used, and it's generally the situation. Interestingly, they're both 6 carbons with 6 hydroxyl groups, and they can be linear or rings. But the fructose, one of the carbons sticks off of the 5-membered ring to make a total of 6. Whereas in glucose and in galactose, the 6 carbons are attached to each other in a cyclohexane type of structure with 6 hydroxyl groups sticking off. Lactose is broken down into a glucose and a galactose. Lactose, galactose, that's the mnemonic by lactase. Next question, question 7. How are sugars and amino acids absorbed from the intestine into the bloodstream? What are the transporters? What am I talking about? The answer is that there's a sodium, glucose, or galactose cotransporter that transports our sugars after they've been broken down by amylase and then by lactase, maltase, or sucrase into monosaccharides into the interior of the epithelial cell. How does it get from there to the lumen of the capillary, where it's transported to the liver? Transported to the liver? Yes, transported to the liver by the hepatic portal vein. Well, this little black dot refers to facilitated transport, and we've talked about this before. There are proteins that transport glucose and galactose. They can get saturated if the concentration gets high enough. We use that at the very beginning of the course. They're called GLUT transporter. There's many different types of them. GLUT4 is found in muscle, but in intestine we have a different GLUT transporter that facilitates the diffusion out of the basolateral membrane so that it can flow down its concentration gradient into the interstitial fluid, down the concentration gradient through fenestrations in the capillaries to get into the plasma where it's dissolved the whole time and can be transported first to the liver and then from the liver to the rest of the body. Where does the sodium go? Well, it gets pumped out the basolateral side as usual. Potassium gets pumped in. Fructose is different. It has a facilitated transporter in the apical membrane, diffusing down its concentration gradient. Also at the basolateral membrane, it gets into the bloodstream. Now you're going to need to know the differences between endopeptidases and exopeptidases, which is where they hydrolyze proteins, the middle versus the ends. You're going to need to know the names of those in the stomach, pancreas, intestinal wall. So we've talked about trypsin, cleaving chymotrypsinogen by clipping it in the middle somewhere. So it is an endopeptidase. You're going to need to know that. We've already touched on that stuff and on the transporters for the amino acids. How the amino acids, once they get clipped off the ends by the exopeptidases, we've talked just now about how they, in the previous question, how they get taken up, but we didn't see that yet. But many of you answered correctly that it's through sodium amino acid cotransport. So we've touched on these things already. Let me move on to another aspect of protein absorption. There are also apical transporters for di- and tripeptides if they haven't been broken down to single amino acids. What is the cotransported ion in the apical membrane and the counterported ion in the basolateral membrane for the di- and tripeptides? There's also a name for the type of transport used to transport even larger proteins and larger peptides. And this is especially important for newborns where they have quite permeable epithelial linings, so they can take proteins up into the body, and it's important for setting them up to have an immune system that knows self from non-self and what antigens there are in the environment. So what are these cotransporters called, and what's the type of transport used to transfer even larger peptides?
Dr. Busey?
Mm-hmm.
I've had some students email me asking about the format of the final and how comprehensive it is. I don't know if you want to answer that in the next couple of days. But.
You bet. Okay, so endopeptidases clip peptide bonds in the middle of a peptide or protein, whereas exopeptidases are either aminopeptidases, which cleave off the amino terminal amino acid, or carboxypeptidases that cleave off the carboxy terminal amino acid, so that you have simple amino acids. And through this means our proteins are broken down mostly to amino acids in the intestine. Trypsin and chymotrypsin being the main endopeptidases, and there being aminopeptidases and carboxypeptidases lining the walls of the digestive tract. So that we end up mostly with amino acids getting absorbed through sodium amino acid cotransport. They get absorbed on one side by a cotransporter, the apical side. On the other side, they get kicked out by a counterporter. So the basolateral membrane has the opposite type of transporter in it than the apical membrane, and that really helps. That combination pumps the amino acids into the blood to get to the liver using the sodium gradient. It's a cotransport mechanism. The question here then is, what helps dipeptides and tripeptides get in? Some of that can also get transported through the cell with hydrogen as the co-transporter in the apical membrane and the counter-transporter in the basolateral membrane instead of sodium. So different proteins to transport the dipeptides and tripeptides. Larger peptides can also be transported through pinocytosis through the cell, and that is called pinocytosis on one side, exocytosis on the other is called transcytosis. Transcytosis. Sometimes adults find they're very gluten sensitive. It is currently thought that extra sensitization of the immune system to gluten during the newborn stage, when a lot of gluten can be taken in, might be responsible for gluten sensitivities, and that's being understood better and better. Before we go on to the next question, Morgan asked me to say a word about the final exam, and I promised that within this week we would have that pinned down, so Morgan and I need to still come to some conclusions about it, but my current plan is to have about an 87 multiple choice question exam that we administer somehow electronically. And that it will have it will certainly be available during whatever our class time exam period is, which was never defined by the NAP, but I will not say how broadly until we figure that out. Next question. How do lipase and colipase and bile salts play a role in fat absorption? What apical membrane transporter mechanisms are involved? What are chylomicrons made of? Where and how are they secreted from the basolateral membrane? From the intestines can go. Okay, answer is our triglycerides are broken down into monoglycerides, not to be confused as I did a moment ago with monosaccharides, which are sugars. These are glycerol head groups, which is what the yellow box means. That's a 3-carbon, 3-hydroxide alkane, or polyol is a better word for it, attached through an ester linkage to a fatty acid, plus 2 free fatty acids. The ester linkage has been cleaved by lipase or colipase, so that now we have free fatty acids and monoglycerides in the chyme. The triglycerides, monoglycerides, free fatty acids tend to glob up into lipid droplets, which then get coated with bile salts with their polar head chains on one side. They have cholesterol in them and breakdown products from heme, from the porphyrins that make up the hemes. And they tend to coat all all 360 degrees around the whole spherical droplet to emulsify the lipid. This lipid is mostly triglyceride at first, as the bile is secreted into the duodenum from the gallbladder and coats the fat. But as the lipase and colipase, the phospholipase and colipase, break down the triglycerides, they get to be more complex. They have cholesterol as well. They also get smaller to the point that you might call them micelles. And eventually they get taken up by the apical membrane of the epithelial cells line in the intestine. The cholesterol is transported through cholesterol transporters. The fatty acids and monoglycerides can diffuse through the membrane because they're so hydrophobic. Interestingly, they get taken up by the smooth endoplasmic reticulum and recombined into triglycerides. The cholesterol also gets taken up into the smooth ER, the Golgi, and then secreted from the tips of the Golgi apparatus as chylomicrons. So chylomicrons are like organelles with a lipid membrane surrounding a fatty droplet composed of cholesterol, triglycerides, and proteins. And these get exocytosed into the interstitial fluid. But they're too big to get into the capillaries. They get through the flap valves of the lacteals. These are the capillaries of the lymphatic system. Now, the fluid in the intestine we call chyme, and the fluid in the lacteals we call chyle. So they are now forming the milky fluid, which is why the lacteal is named that, the chyle. It's going to go through the cisterna chyli, you might remember from anatomy, in the high abdomen, up through the thoracic duct and get dumped into the subclavian vein, and from there to the superior vena cava. Okay, so our fat gets to the veins via the lymphatics that have milky lymph in them called chyle. But these chylomicrons are created first in the epithelial cells and then exocytosed where they can get there. Question 10. On to the liver. 5 questions about hepatocytes in the liver. The liver is a magnificent feat of engineering comprised of long, I can't think of the word I want, strands of hepatocytes that are exposed to the blood coming in from the intestines, as well as oxygenated blood from the aorta. And that blood gets processed as it passes by these hepatocytes. How do they process toxins, drugs, and alcohol? What blood vessels bring these things in? What are the breakdown products of heme metabolism? And what vessels take them out, breakdown products? In what form is glucose stored in hepatocytes? What proteins that we've talked about previously in the class do hepatocytes produce? Go.
Hey, Dr. B, I have a question about triglycerides from one of the students. So why does the body break them down if it's just going to reform them later on?
They need to be broken down to be dissolved enough in the plasma so that they can then diffuse through the cell membrane. Because triglycerides would just stay clumped up and have the bile salts around them and would never get free into the plasma to be able to get through the membrane, the apical membrane? Great question. Okay, the many answers are first, the second question from the oxygen. So it's diagrammed as blue here, and atomically it's coming in at the corner of a hepatic lobule, which is a hexagonal lobule, and there's a hepatic triad consisting of the hepatic portal vein, which gives which leads to a sinusoid. Spicules was the word I was trying to think of before. Spicules of hepatocytes. These sinusoids pass among the spicules of hepatocytes and aim for a central vein that goes to the inferior vena cava through the hepatic vein, the outflow from the liver. The abdominal aorta sends a hepatic artery to bring oxygenated blood to those spicules as well. And the blood gets processed by the hepatocytes through a variety of enzymes, was the word I was looking for. Enzymes that break down these drugs, alcohol, and toxins. They are going to be released in the next slide into these sinusoids called canaliculi, little canals that run between the spicules, and the products are going to become the bile salts we were talking about in the previous slide. These bile ductules will all gather together into vessels that lead to the gallbladder, where the bile can be stored, common hepatic duct, the common bile duct, and then stored in the gallbladder. When the gallbladder is, by a smooth muscle contraction, squeezed closed, it squeezes the bile out. Which was the hormone that activated that? CCK. The bile is squeezed out to enter into this spot of the duodenum. This is the duodenum right here, the little C-shaped hook coming out of the stomach, with the pyloric sphincter here, squeezing the chyme into the duodenum, stimulating the release of CCK via the bloodstream to stimulate bile release into the duodenum to help emulsify the fat. This hepatic portal system is a second set of capillaries, because the first set was in the digestive tract itself that we were looking at before, picking up the sodium amino acid, sodium glucose, co-transported nutrients, and bringing them up to the liver. The chylomicrons, the fats, aren't picked up here. They're taken back to the heart, where they're distributed via the lungs and eventually through the thoracic and abdominal aorta. So what are the different mechanisms that the liver uses to detoxify toxins? Well, it breaks down some toxins into small molecules that end up as small solutes in the intestine through the bile, or the urea and other small solutes get out through the hepatic vein, and then they're processed by the kidneys, which secrete the toxins. urea is one of the breakdown products of glucose and metabolism. The hepatic artery is bringing in breakdown products from the spleen of the red blood cells, where the hemoglobin is getting converted into the protein globin, and the heme groups, which then are converted into bilirubin and secreted into the intestine as bile. The bilirubin is also processed by the through the hepatic artery through the kidneys to make our urine yellow, you'll recall. Proteins that are synthesized in the liver and secreted via the hepatic vein into the plasma include albumin, the clotting factors we've talked about, the fibrinogen, and the thrombin. Also, angiotensinogen that is cleaved by renin to make angiotensin 1, which is cleaved by angiotensin converting enzyme to make angiotensin 2, to stimulate the adrenal gland to secrete aldosterone to stimulate sodium reabsorption in the distal convoluted tubule of the nephron. One other thing that we'll talk more about in the future is the processing of vitamin D. And we have talked in the past about how the anterior pituitary gland releases insulin-like growth factor to stimulate somatomedins, to stimulate body growth. It secretes growth factor, which stimulates somatomedins, also known as insulin-like growth factor. Question 11. We have this liquid chyme, but we poop out feces. How is the fecal consistency fine-tuned by crypt cells? If CFTR is non-functional, you have constipation. If it's phosphorylated, it gets overactive, you pump out too much chloride and you get diarrhea. So either constipation or diarrhea. What disease results in severe diarrhea by secreting a toxin that activates a G protein, causing high cyclic AMP production, leading to CFTR phosphorylation, and therefore hyperactivity of CFTR? Go. Answer is, well, first, before we talk about the crypt cells, let's note that the colonocytes in general, meaning the colon cells, the epithelial cells lining the colon, are going to constitutively reabsorb sodium and water through sodium pumps in the basolateral membrane, chloride channels that pull water with the salt reabsorption to dry up the chyme that is squirted into the intestine at the ileocecal valve after passing through the small intestine. And that the chloride and sodium that are being transported this way into the interstitial fluid are coming through sodium channels, sodium chloride co-transporters, sodium hydrogen antiporters, chloride bicarbonate counterporters, potassium channels, letting potassium out. So all of this is drying up the feces, water salt following via this paracellular path, potassium being excreted in the process. And then the fine tuning is done by crypt cells, which have cystic fibrosis transmembrane regulator channel in the apical membrane to let the chloride out, which is being brought in through secondary active transport, depending on the sodium gradient produced by the primary active transporter, sodium potassium ATPase. The chloride then getting into the lumen of the colonic crypt and drawing sodium and water to keep our feces from getting too dried out. In the absence of CFTR, if it's been mutated and you have cystic fibrosis, you might have constipation. You would have constipation constantly. Another problem they have to deal with, clogged up pancreatic ducts, clogged up colon, and clogged up lungs, and some sweating problems are constant battles for cystic fibrosis patients. What happens if CFTR is overactive? Cholera has secreted a toxin that stimulates cyclic AMP production in the cell, phosphorylating CFTR and making it overactive. Cholera can be deadly. Last question.
Gotta go fast. It's already 9:52.
Cytokines from gut-associated lymphatic tissue, GALT, is blamed for inflammatory bowel disease, which is a chronic diarrhea problem. Shigella and Salmonella are bacteria that cause diarrhea by commandeering M cells in the GALT. What about vomiting? Where is the stomach contraction reflex involved in vomiting? Also the abdominal wall contraction reflex. Where is that initiated and why? Answer is back up in the brainstem. There's a medullary control center which detects drugs, cytokines that get into the blood, sometimes pain, pharyngeal stimulation if you poke the back of your throat, motion if you have motion sickness, and even psychically, you can activate this medullary control center to cause nausea and vomiting. That's it for today. We will see you again on Wednesday. Keep up the good work. You're all doing great. I appreciate how hard this must be for you and wish you the best. Thank you. Goodbye, y'all. So Morgan, I'm still here, and Xander seems to be here. A few haven't logged out yet. Have you stopped the recording?